So, thank you for inviting me to speak today. It's actually a little bit of a circle in my life because in the 1990s I visited your patron, Veronica Van Henigen in Edinburgh, to get some career advice about doing genetics and ophthalmology, and that was in the late 1990s. And so my career's been slightly strange. So I've trained in corneal disease. I did my thesis on a different genetic disease that affects the cornea. I went to the Sick Kids Hospital in Toronto and spent 2 years there doing genetics. I then went to Belfast for 9 years, and I do cornea and glaucoma in Belfast. I did cornea and glaucoma in Belfast, and now I'm back in Liverpool. And so my life split 50% in the clinic and 50% in the lab, doing research into genetics and molecular biology. And so it was really Veronica and the Aniridia UK that asked me to discuss the topic of this talk, which is actually quite technical. So I've cut it down a little bit because Alex covered some of the things that I was going to include. So I've made it slightly smaller. And I suppose the idea behind this talk is that some of you may be aware that there was a drug treatment published for Aniridia recently in January 2014. And really it was to go through that study critically and explain the sort of genetics and the potential for any sort of molecular treatment for Aniridia, and put that paper in its context, because I know some colleagues of mine from around the UK have emailed me to say, you know, patients have walked in with this paper and asked for this drug treatment. So it's really just to put the paper and the treatment in some sort of context, and then also to talk about some early work that we're doing in Liverpool on Aniridia and in different molecular treatments for Aniridia. So I'll try and make this digestible, like your lunch. I think we've got a little bit of time, so if it's technical and it's difficult, just stop me and I'll try and explain it in a better way. So you've all got your pictures of the eye in front. We've heard some terms today, and I know not all of you are ophthalmologists. The main things we're going to talk about really are the cornea, which is like the front, the cornea, which is almost like the front windscreen of your car, okay? And then you've got the iris behind that, and then the lens behind that, which is, we're talking about cataract today. And then at the very back of the eye, like the film and the camera, you've got the retina. And those are the structures that we're going to discuss. So if you look on your website, last year there was a very elegant talk that covered all aspects of the ophthalmic treatment of Aniridia, and it's on a video that you can watch. So I'm not going to really say very much about that side of things. But Aniridia was originally described because it affects the iris, and an in medicine means absence of the iris. But there is a range of iris defects from complete absence to partial absence to minor abnormalities. But we know that Aniridia is not just a disorder of the iris. It's called a panocular or a whole eye disease or a disorder. And so it can affect the eye in different stages of life. So from birth, in the first year of life, in infancy, in childhood, in your teens, and then into adulthood. And some of the features don't become manifest until you go through those different phases of life. And the major ocular effects, it affects the cornea, which we've heard about this morning, glaucoma, raised pressure inside the eye, which causes damage to the optic nerve, cataract, dislocated lenses, the iris defects, foveal hyperplasia and optic nerve hyperplasia, which are usually from birth, and they impact by causing problems with the vision and nystagmus, and that's really a sort of a developmental problem, where some of the other problems develop as you get older. And it's just a slide to show what foveal hyperplasia means. It's just a technical term that the part of the eye that's most sensitive for vision is underdeveloped. So the gene that causes Aniridia is called PAX6, and PAX6 is a gene which regulates the development of the eye. So it's a master control gene for eye development, and so it's been conserved across many species, from flies up to humans. And if you have abnormalities in PAX6, it leads to developmental disorders in the eye. So that's the main genetic cause for Aniridia. So just a little bit about genetics. I'm going to go through this in a little bit more detail in a moment, just to explain it a little bit better. So you could have Aniridia if you have a family history, and it's inherited in an autosomal dominant fashion. I'll explain what that means, and I'll explain what a heterozygous person is. But that's in about 2/3 of cases. In another third of cases, you can be born without a family history. So this could be a new mutation in PAX6, or it could be because a piece of the chromosome, the chromosome would be equivalent to thinking of the UK. If we imagine Scotland as part of a chromosome and it decides to leave the United Kingdom, it breaks away. So it doesn't just affect Scotland, but it will affect the different cities within Scotland. So if you imagine PAX6, we'll say as Edinburgh, because Veronica's here, that's the gene, whereas Scotland would be the bit of the chromosome. It wouldn't be all of the United Kingdom, but a small piece of it. And if that happens, you can lose other genes, and that's the genes that are associated with the Wilms tumour, or the adrenal gland tumours, or the kidney tumours that you see. So if you have a deletion of the chromosome, you're likely to get other medical problems. So about 85% of cases it's due to a defect in PAX6 gene alone. So it's just a problem in Edinburgh, it's not a problem in Scotland. So the way genes are inherited, we all have 2 copies of every gene. One we get from our mother, one we get from our father. And if you take a patient with Aniridia, so the red here is the affected or the abnormal PAX6, and that's their partner. So it means that there's a 50-50 chance that they will have a child affected with PAX6. You can see that they can either give the normal PAX6 copy or they can give the abnormal PAX6 copy. So that's heads or tails, so 50-50. So every pregnancy, 50-50. Now this is the PAX6 gene, so I need to explain this so that I can explain the therapies. So the best way I can explain a gene, and I'm going to use 2 analogies here. This photograph, or this picture, shows a gene like a bead necklace, okay? So the beads on the necklace are the bits of the gene that make its product, and the little bits of string between the beads are little intervening bits of the gene. So all the beads have to be chopped out, placed together to form a protein, which then has some effect in the human. Okay, so that's the bead necklace analogy. The other thing I'd like to explain, because we're going to talk about things like stops and full stops and things like that, missense and things. If you imagine a gene as like a book, okay, at the front cover is the bit that you open, so that's the switch that turns the gene on. And then you've got all the chapters and the paragraphs within the book. And then at the very end, you'll come to usually the end, and that's called a Stop-Codon. I'm going to show you what that does in a moment. And then you've got the back cover of the book, which we know in genetics also has a function for the gene. So I'm going to talk about those different aspects of a gene. This is PAX6, and the little black boxes are the beads on the necklace, and the black thin line is the string. Okay, and so those little lines get chopped out and the beads get put together, and it forms this protein called PAX6, which you heard about in the talk just before lunch. So when Veronica used to look for those genetic defects, you had to sequence your genes, and this is what happens when we sequence your genes. We see these little peaks for your genetic code. So the genetic code isn't like our alphabet. We don't have 26 letters, we only have 4 letters. It's G, A, T, and C. So every gene in your body uses those 4 letters. So it's like reading a book and you're only allowed to write in G's, A's, T's, and C's. And what we've got to do when we look for a genetic defect is find that typo, find where the G isn't a G or the A isn't an A. And so that's why it's quite difficult. And in the early days of genetic sequencing, that took a long time, and it was quite labour intensive. You had to go through a whole book and find that one typo where a G was no longer a G, it was something else. And just to show that, this is someone who's TT. So they've got 2 T's, and then here the mutation, that's a C to T. So there's a little blue hump with the red, and that's the mutation. So that's what Professor Van Henigan used to have to try and find in your genetic code to determine whether it's a PAX6 mutation. And that's what we still do in some respects today. So this means that the person here is what we call heterozygous. So heterozygous means they've got 2 different sort of copies of the gene. So the only picture I could find to explain heterozygous was a heterozygote, which is a technical term we use in genetics, but it's not that type of goat. But basically, we have a black and a white goat, and they're sort of 1/2's black, 1/2's white. So imagine they've got 1/2, the black bit comes from the mum, the white bit comes from the dad. So in a heterozygous mutation, basically you've got one copy which is, let's say, black is the way the goat should be black, and you should have two black copies, but you've got a black and a white copy. So that's what we call a heterozygote. So it's like a mixture. Okay? So there are different ways we talk about mutations. So the one that I explained about the typo in the book, basically here, hopefully that's pretty, sorry. We've got a CCC which codes for one of the parts of the little bead, and here we've got someone who's got an ACC, so that's called a missense mutation. It's like writing Bob and then putting a T at the end and have bot instead. So it still makes a bit of sense, but it's not quite right, but it doesn't damage the sort of structure of the sentence. The next type of mutation, this one here, you get actually the end written in the middle of the book. Okay, so instead of you being able to read the book right to the very end, you've got the end bit stuck somewhere in the middle of the book. And so that stops the gene functioning and makes it smaller, and makes the protein and it makes smaller. Okay, so that's a stop-codon. And sometimes you get a part of a word or some letters taken out of the word, and then all the words move across. So it turns into like a gobbledygook after that. So it's like if you wrote your address down, and I told you to take 3 letters out of the middle and keep all the places of all the other letters, then suddenly it would just turn into gobbledygook. And what happens then in your body is that gobbledygook reads and reads and reads until it finds a stop. So basically, if you read the book, all the words would just be gobbledygook, and then suddenly there'll be a stop in the middle of the book again. So those drug treatments that I'm going to talk about try and affect some of those stop codons, those stops that appear in the middle of the book. So you saw this slide a little bit before. So you've got the beads from the gene, okay, and then those beads are read into what we call a messenger sort of gene, and then that goes out into the cell or into your body, and then that's read, and that makes a protein. Okay, so that's like your wife opening up Jamie Oliver's cookbook. She tells you to read it to her, and then she makes the meal. The meal is the protein. You reading it, the chapter to her or the book to her is the message, and then the actual code in the recipe book is actually the genetic code. So those 3 steps to generate a protein: the book, someone to read the book, someone to make the meal. So what happens when you have these stop codons is that when you're reading your cookery book or whatever book and there's a stop where there shouldn't be a stop, your body's designed to deal with that. It has a sort of a safety mechanism because it doesn't want you to damage the protein, or it doesn't want to make a protein that's abnormal. And so I've shown that here by this little Pac-Man thing. So here we've got the gene. Okay, there's its start. There's the little beads with the bits of string. There's where the stop should be at the end of the book. And suddenly there's a stop in the middle of the gene. Your body's designed to deal with that, so it basically eats that, and it won't let it be made into the meal. Okay, so that's you reading out the cookery recipe, and then your wife clubbing you on the head with a frying pan. You're not going to make the meal. Okay, so your body deals with that mutation. And that's a sort of a natural thing that's in all our body to sort of deal with genetic mutations of this type. So we call it a PTC, which is a premature termination codon or a premature stop codon, basically a premature stop in the genetic code. And so the reason you get aniridia, the mechanism, is that because you get 2 copies of every gene, so you should make a protein from your mum's copy of the gene and a protein from your dad's copy of the gene. So basically you've got 2 copies. So you've got 2 chicken breasts to cook your meal with. Well, in aniridia, one of the chicken breasts gets degraded, so you only have one of the piece of the protein. And just to show that, so if you look here, this is a normal patient, so they make 2 copies of the protein because they've got 2 copies of the normal gene. But if you've got aniridia, you only make 1 copy of the protein, so you've only got 50% the amount of the normal protein that you need. And it's that dosage effect that's one of the reasons why you get aniridia. So you only have 50% of the normal amount of PAX6. So the idea behind these treatments are, how can you reverse that how can you make like 100% of the protein? Is there anything you could do with a drug treatment or a molecular treatment that could make more of the protein? So this is the START treatment. So this was the acronym coined from that paper. And I'm just going to run through what they did and what they found and sort of how the thing works and some of the considerations about that type of treatment. So the way the treatment works is it tries to trick your body. So here's the person with Aniridia with the Stop-Codon in the middle of their book. It tries to use a drug to hide that Stop-Codon so that the protein still gets made. Okay, so it just tries to trick your body into not degrading that, so you don't get the Pac-Man eating that up. It gets made into a protein. And so this is the drug. So it's called PTC124, but they called it Autolurin, which is a drug which causes this Stop-Codon read-through. So does an antibiotic, which I'll talk about in a little minute. So there's a very common antibiotic we use in medicine called gentamicin, and it works in a similar way. So this drug was developed for other genetic diseases, and this group in Canada thought they should try and apply it to Aniridia. So this was the paper published in January 2014. I'm just going to run through what they did. So the first thing is they looked at 6 mice, okay? So this was 6 animals. And the animal had one of those stop-codons, and it was a natural, well not a natural, but it was a known model for Aniridia that affects mice. And when you look at the mouse eye, so on the top there we have the normal mouse eye. You can see the mouse has a huge lens for the size of its eye, which is this big red blob, like a cherry. And it has a cornea, and then this layered sort of thing at the back is the retina, and then that's the optic nerve. So it's just like your picture, but the lens is huge inside the eye. And in the mouse with Aniridia, the cornea is sort of thickened. The lens is sometimes stuck to the back of the cornea or adherent to it. It's abnormal, and then the retina is this sort of folded up mass of tissue. So what they decided to do was to give these mice either gentamicin, the antibiotic, into the whole animal's body, the new drug, the Autolurin, into the whole animal's body, and then they thought, could they just give it as an eye drop? So they tried it as an eye drop, and then they realized that caused a lot of irritation, so they basically blended the drug with some other things. So some of these things are the dry eye drops that you take, some of them are just sort of water and buffers and things like that. And what they showed was that they could actually increase the level of that PAX6 protein. So instead of having the one chicken breast, you actually had 90%, so you nearly had enough protein that should work in the eye. So I'm just going to show you what they found. So first of all, they treated the animals after postnatally. These are like little mice pups. They treated them with the drug systemically, and what you can basically see, I think, in the best picture would be if this is the normal mouse, and then this is the Aniridia mouse. This is what happened when they treated it systemically. So you could see that the retina looked normal, the lens looked normal, and there was still some changes in the cornea, but overall the eye looked generally more normal than it did without the treatment. And they also tested a mouse which didn't have that Stop-Codon type mutation, and it didn't really have an effect. So they thought this was a sort of a specific response to the treatment. So then they tried, well, they did deliver it topically, and I'm just going to show you what they found when they delivered it as the eye drop with the buffers. And again, they treated the animal after birth, and so I think the best pictures would be these. So that's the wild type animal, that's the one with the topical eye drops, and that's it without the eye drops, which is again, it's quite dramatic. And so you can see why there was a lot of interest in this as a form of treatment, because it almost looks like a perfect form of treatment. Then they showed the cornea. So they took there's the lens, they just took the cornea, the bit at the front of the eye, like the car windscreen. You saw the different layers of the cornea in the earlier talks. And so this is the control animal, the normal animal, and then this is the animal with the topical treatment. And there's a few things here that I took issue with. They said in the paper that this was the same as this. But the skin on the front of the eye, to me, doesn't look entirely normal. And there are also, the tissue here doesn't look entirely normal. And they also looked at the thickness of the cornea, and then that was enough for them to publish that this was affecting the corneal disease that Alex showed you before lunch, which I'm not entirely convinced about. They also looked at the retina, so the film at the back of the eye, and they showed an effect in the retina. And again, that's slightly unusual to me because retinal degeneration in that way isn't really a feature of aniridia. So foveal hyperplasia is, and optic nerve hyperplasia, but you don't really see a retinal degeneration normally in someone with aniridia. So there's something different about the mouse than the human. They also looked, and they didn't actually put this in the actual bit, the main bit of the paper, they put this in the bit that you have to go on the web and, like we call it, the extra information or the supplemental information. But these are the animals treated with systemic antibiotics. This is an antibiotic that we used to use long ago for different infections, and they could show rescue of the eye features with that antibiotic. They didn't look at it as a topical treatment, but we do use gentamicin eye drops in clinical ophthalmology to treat infections like conjunctivitis and corneal infection. So this is a drug that we use normally in medicine, but they didn't actually show, the topical treatment versus their new drug of this old drug. So I'm just going to break down the considerations then from this paper. So I think it is interesting, and in some respects it could be a major change in how we think about aniridia, but I think there's still some unanswered questions. So I'm just going to go through the paper-specific unanswered questions, and then about the actual molecular approach, because these things, you read the editorial for them, and you look at the BBC website, and there's always a bit of spin about all of these things. So I think the first thing is it was based only on a mouse, and it was only based on 6 animals, and the mouse only had one specific mutation. 2, the mouse eye is different from a human eye. So mice don't have a fovea, for instance. So there are some intrinsic differences between a mouse eye and a human eye, and that's why people are working on human models of aniridia to look at treatments and things. I don't think the corneal histology as a corneal person was that elegant in terms of whether it was entirely normal. They looked at thickness, which is can be a little bit difficult to characterize, and they didn't look at other features of that would be associated with aniridia and keratopathy that we talked about. So I don't think the corneal, what I call endpoints or treatment points, were that well characterized. As I said, retinal degeneration is not a feature of aniridia in humans normally. I think the comparison groups weren't entirely fair. I think they should have looked at topical gentamicin, which is an antibiotic we use in ophthalmology, versus their topical treatment. There are some risks about using gentamicin. It's quite toxic to the cornea, and so in aniridia you've got to be careful that you're not causing toxicity that might make things worse, but I think they should have looked at that. And I also think that before they did their blend with the drug, it was quite toxic to the mouse eye, quite irritating, and I think that would have to be carefully addressed if you were going to try and use this in a human, because obviously the aniridic cornea and the surface of the eye is not naturally healthy, and then if you give a drug that could make that worse, then you could make things maybe worse rather than better. Okay, then about the actual strategy about using this. So we don't really know the mechanism fully about how some of these drugs work. They only work for mutations where the stop-codon is what we call in frame. That means that the rest of the book reads normally, you just have a big stop in the middle of it. So if it causes that gobbledygook type thing, it wouldn't necessarily work for that. So you still need to know the genetic basis of aniridia, so that there's still work needs to be done in that area. You need to know the mutation, and also how many people would be potentially benefit from this depends on the mutational spectrum in aniridia. When you look at the literature, some mutations in studies respond better than other mutations. So they only used one mouse with one mutation, so you don't know whether that's applicable to all different people who could potentially benefit from the treatment. It relies on the little Pac-Man thing I showed you not working properly. So if he gobbles up the message before it gets told to the cook, this treatment won't work. So that mechanism has to be inefficient. So you're relying on something being inefficient for your treatment to work, which is a little bit of an assumption that you make. And these drugs have been used in other common genetic disorders like cystic fibrosis and Duchenne muscular dystrophy. And the mouse data was encouraging, but when they went to human trials, it didn't really have the effect that they hoped. And so there were lots of trials that have been started and given up on this treatment. Recently there was one on Duchenne muscular dystrophy that showed a little bit of benefit, and it was in terms of walking speed. So really when it comes to the clinical trial, they haven't shown the promise that they might have shown in the laboratory. And it's not a specific treatment, so it could potentially affect stop codons in other genes in your body. They have looked at some of those, or sometimes if you have a stop codon in a gene, it could cause a negative effect on a person, and then if you use some drug to read through that, it will affect every gene in your body, so it's not specific for PAX6. So there's a few things I don't think that have been quite ironed out. So I'm going to stop there. A little bit of promotion for Liverpool. So I've got in Liverpool quite a good nidus of people. So Sarj Ahmed was in London, and he worked with Alex Shaw on stem cell treatments, and also in Newcastle. And he's just been awarded a funding for 3 years to look at how the stem cells in Aniridia, you could hear that Alex said that the stem cells don't survive long in their treatment studies, and some of that could be because you reject them. It could also be how the stem cells interact with the cornea, so we're looking at that. And we've also got expertise in genetic eye disease, corneal disease and glaucoma. So we've got a good nidus of people with sort of complementary interests. And there's Liverpool. Looking bright and beautiful, as ever. So I know that was very hard to explain that to you, but I just know that people are, maybe particularly if you've got younger children, you'll see that paper and you'll think, really I want to use, have this, maybe for my child, but I think you just got to be careful that the studies are done properly and that it's safe to use. And I always think it's not, it's important not to build up people's expectations if you can't deliver anything. Okay, so thank you very much, and I'm happy to take questions on this or whatever you feel like.
I'm in communication with Professor Gregory in Canada in regards to clinical trials and what would you advise there?
Well, I spoke to him recently before I came here, and I haven't spoken to Martin Collison because I sent, I wanted to talk to him about what he felt about the mouse data. But Kevin told me that the trial in the UK isn't at the moment going to go ahead, but he is trying to start a trial in Canada and the US in a small number of patients. So I don't know what sort of regulatory things that he has to put in place to do that, but that obviously will be decided by the FDA in America.
But the full time at the moment, I'm in the process of getting my mutation checked. And then I'm supposed to be getting that details to Professor Moorfield.
Okay.
And that's as much as that.
Well, the latest update I got last week was that wasn't going to go ahead. But I don't know why. He didn't explain to me why. I re-emailed him to ask him why, and he didn't come back to me yet. But there are some issues about it. So you have to have the right type of mutation. It depends where it is. It depends whether the gene is still read properly. And then there's those other issues that I mentioned. I'd really like to see some better data. I think this sort of treatment I could see being helpful potentially for the corneal disease, potentially more than any other part of the eye, because the foveal hyperplasia and the optic nerve hyperplasia happen in utero. So so really you're looking at the corneal problems. And to me, that histology didn't look quite. If I reviewed that paper, I'd be worried about that histology. And also, I think you need to show what it's doing to the stem cells. They didn't show any data like that. So I think it's difficult. Veronica?
I just also want to emphasize that, I've done a lot of mutation analysis, and we keep a database. There are very few people really who have the right mutation for that therapy even to be considered.
Yeah, so it's a small subset of people with Aniridia that could potentially benefit, depending on how effective it is and how safe it is. But the way the paper was written, it's like, it sounded like this miracle. Not the paper, but there was an editorial that sounded like this wonder treatment. It happened postnatally that the retina suddenly formed. Well, mice are born almost blind, and then they develop, because they're nocturnal. They develop their vision sort of after birth. Whereas we're dealing with humans where most of our visual structures are very early in life, that they're formed in utero. So it might not. I think it's interesting. I wouldn't you know, but I think it has to be taken, you know, carefully.
Well, I was looking at it, not for as much for my own staff, but if I could help in research for future people with it.
Yeah, I mean, and there's things you can do in the laboratory to look at the stem cells. So I didn't really go into it here, but some of the groups we work with, you can take cells and deprogram back to being a stem cell, and then you can see if they can still make a cornea. You can do work like that with these sorts of drugs and see whether they still have the potential for the stem cells to make a cornea, make sure it's very, you know, it's safe to use and things. So, but yeah, I mean, it's one strategy. I think, you know, there'll be different strategies for different clinical problems. Right.
It is a drug that's being used being tried, that would be good, but I don't know whether at a clinical level.
No, there has been clinical trial. Yeah, and Duchenne muscular dystrophy. And the company's share price has gone up and down because there's always great expectation, and then the data didn't come out in the clinical trials, and that's why they've tried all these different diseases. And I could show you tables where it was like trial discontinued. There were a few safety issues, but they don't really go into those. I haven't been through every single trial. But the recent one, the Duchenne muscular dystrophy, the European Union were looking at granting orphan drug status to this molecule because it showed some improvement in a child's walking speed. But I don't know what loops Kevin or Professor Greg Evans have to go through in the US, but you know, I think you need to understand what it's doing to the stem cells a little bit better, really. Okay, thanks very much.
Thank you very much.